All right, 12s, Mr. K here in the science bunker. We're going to be talking about in this video about capillary tissue fluid exchange. So what does that mean? Well, this is how gases and dissolved amino acids, oxygen, glucose, those things and waste get exchanged in the capillaries. We're zooming in on the capillaries. Get exchanged with the tissue cells, with your tissue cells. So let's zoom in. Here is an image of capillary. There's the capillary, and you can see the capillary is one cell. The diameter is one cell thick. There's some red blood cells in there. And some other stuff. I don't know if you can see in the video, there's the little blue dots. Those are plasma proteins, dissolved proteins. Remember, there's proteins inside the plasma. These would be albumin will come into play here. So these are proteins that control the osmotic pressure that keep the blood hypertonic. I'm going to tell you in a moment why that's important here. So, in this image, what you see is this, at this end, this is the arterial end. So this is coming from the heart. This is closer to the heart. This is a capillary in the tissues, in a capillary bed. Most likely this capillary would be a little bit longer. And this is the venous end. So this is heading out to the veins, and venules, and then veins, and then going back to the heart. So remember, blood pressure drops as you enter the capillaries. You lose the influence of the heart, the pressure, the contraction of the ventricles as you enter the capillaries, because the capillaries are branching out and out more, and the total cross-sectional area, the total cross-sectional diameter of all the capillaries is greater than of the arteries, so blood slows down and the pressure reduces. And this happens somewhat gradually as we go from the arterial end to the venous end. Now, things you need to know also, the capillary wall is porous. It's got little spaces, little pores in between it, little holes in the capillary end. They're small. Red blood cells can't pass through them. Blood proteins can't pass through them. That's too big to let blood proteins, so globulins, pass through. But smaller molecules like amino acids, glucose, water, dissolved gases like oxygen, those can pass through those little, it's a semi-permeable membrane. They can pass through those openings. Now, at this arterial end, what is happening? You see these big arrows here. So, number one, something to know, there's osmotic pressure. So the extracellular fluid, that's what this is called. It's not this, so there's the cells, and this is the tissue fluid, or the fluid around the cells, is actually pretty watery. And what's happening is because there are these plasma proteins and red blood cells and all sorts of dissolved goodies inside the capillary is hypertonic to the outside. Remember all that stuff that we did in the membrane, to the outside, to the tissue fluid. So there is a concentration gradient going out and an osmotic gradient going into the capillary. So water wants to go into the capillary. At the arterial end, it's constant. This gradient is constant, and one of the things that keeps it constant are these blood proteins. That's what those albumins, those dissolved globulins, are for keeping the plasma hypertonic. So what happens, so we've got a fairly constant osmotic pressure of water wanting to go into the capillary. 21 millimeters of mercury of pressure. However, at the arterial end, water does not go in to the arterial. It actually gets pushed out. And the reason it gets pushed out because of blood pressure. So pressure here is still high enough, even though we're entering all those branched capillaries, pressure is still high enough. The contraction of the heart, that's what's happening, is the pressure from the heart is still high enough here that it is greater than this osmotic pressure. Pressure here is 30 millimeters of mercury, which gives you a net of 9 millimeters of mercury out. So what happens is water is squeezed out of the capillary through those holes. It's squeezed out. Out it goes, out into the tissue fluid. And that water carries with it, because dissolved in it are amino acids, glucose, nucleic acids, some fatty acids, and salts as well are going out here. Now, cells that need that stuff are going to gobble it up. We're going to take it up. Cells that don't won't. They don't. But, you know, glucose will just pass. Those things will go in. Water will go in as well. Cells that need water. Oxygen. So these red blood cells are carrying oxygen. And in here, we'll learn about this in the respiration unit. But the oxygen is being carried on the red blood cells, on the surface of the red blood cells, on a protein called hemoglobin. And hemoglobin is very sensitive. It has a very thin pH range and a very thin temperature range at which it works. In the lungs, because the lungs, you're breathing in air from the outside, the lungs are a little bit cooler than the tissue cells. Here, there's probably muscle and stuff around here heating stuff up. These cells are using ATP. They're heating stuff up. So the lungs are a little bit cooler. It's like it's like a fraction of a degree. Also, in the lungs, things are a little bit more basic. And when I say they're 7.35, I believe, is the pH in the lungs. Here, things are a little bit more acidic because there's carbon dioxide being carbon dioxide waste coming out of these cells, and it makes things more acidic. Now, so in the lungs, it's 7.35. Here, it's 7.38. That's the difference. And a little bit warmer, like a fraction of a degree or a degree warmer. And that is enough, that difference is enough to change the shape of hemoglobin, that different in pH and temperature, where it just can't hang on to oxygen that well. It starts to denature, I guess, in a way. It actually can hold other things. It can hold carbon dioxide and protons or hydrogen ions. It can hold hydrogen ions, but it doesn't hang on to its oxygen. So as soon as it enters here where it's warmer, it's a little bit more acidic, it starts to lose, the hemoglobin starts to lose its oxygen. The oxygen dissolves into the plasma. There's a concentration gradient because there's not going to be any oxygen in the tissue fluid because these cells have gobbled that oxygen out. So that oxygen is pushed out along with the water and along with diffusion. It diffuses out of the capillary, and all of that oxygen is going to go up. That concentration gradient of oxygen is going to be maintained because all these cells, as soon as that oxygen gets out there, is going to gobble them up. So this tissue fluid will never have oxygen in it, and so there'll be a concentration gradient from if there's any oxygen in here left over, it will always want to go out into the tissue fluid, and the cells gobble it up, as well as glucose and amino acids. And all of those things, they'll just gobble up. So there's always going to be a concentration gradient, and to help it is also this pressure gradient. So that's how the nutrients and stuff are delivered. There's a concentration gradient, and there's blood pressure pushing stuff out along with the water. Now, there's not a net movement of filtering or much stuff in the middle going on. Maybe there's some diffusion going on, but then what happens is now, as this blood has been traveling slowly, slowing down and traveling through the capillary, the pressure from the heart the influence of the heart is dropping. And so this blood pressure is going down and down and down and down until you get to the venous end where the blood pressure is half of what it was, 15 millimeters of mercury. But the osmotic gradient of water wanting to go in, because of the albumins, because of those proteins in there that are making the plasma hypertonic, that hasn't changed. So now we have a net pressure of 6 millimeters of mercury of fluid and water returning to the capillaries. So you're reclaiming that water. And along with that water, any wastes that these cells have excreted are also being brought in. There's also going to be a concentration gradient for the waste, so they're also going to be diffusing in, but they're also going to be helped along by the water. So carbon dioxide and nitrogenous wastes, anything else, are going to be going into there. That's how wastes come back. That's how the capillaries work. Some things to note. 9-6. Less water is coming back than went out. That water is, some of it is going to be used by the cells, and some of it is going to be reclaimed by the lymphatic system, which we're going to be talking about in this unit in another video. So that's where that goes. When you have an immune response, when, say, there's some kind of infection or something going on here, white blood cells, leukocytes called basophils, will end up coming here. And basophils, what they do is they secrete histamines. You may have heard of histamines. Antihistamine is an anti-inflammatory medicine. And what histamines do is they open the pores of this capillary. They open the pores of the capillary. So when there's histamines present, the pores are open. So more water pours out into the extracellular fluid. The goal of that is if there's an infection here, there's some kind of bacteria, the goal is to wash it away, to increase the opening the pores in the capillary. That blood pressure is going to be pouring more water out and trying to wash the bacteria out into the lymphatic system. The lymphatic system has lots of white blood cells in it. Lymphocytes, you may have heard of lymphocytes. So trying to wash it out here and basically send these pathogens out of the tissue and to the wolves in the lymphatic system, those white blood cells that are going to attack those pathogens. And that's why inflammation is the tissue swelling. Because there's so much water pouring out, it's getting in between, it's filling up, and you become swollen. Because it's filling up with water. And that's thanks to the histamines that these basophils, these white blood cells, are producing. So when you get a mosquito bite, that lump is, there's histamines there, your immune system is responding to the saliva of the mosquito, essentially, the stuff that's in the saliva of the mosquito, and it is swelling that area. Over time, as the pathogen is washed away, the need for histamines goes down, histamines stop being produced, and the swelling will go down. Anything else to say about this little bit? No, I think that is it. Here's another image. This is from the textbook, so you can see here filtration is going in. So arterial end, more is going out. Reabsorption at the venous end. Sort of no net movement at in the middle there. Anything else I'm forgetting? No. Okay, there you go.